the coding metaphor, and the mythical genetic code. Only a small fraction, around 1 to 2% of typical animal genomes, code for amino acid sequences of protein chains. In other words, only a small fraction consists of codons that are transcribed into RNA, spliced together during RNA processing to make mature mRNA, and translated into amino acid sequences at the ribosomes. Many DNA sequences, called introns, are removed, spliced out, during processing of RNA. The retained portions found in mRNA are called exons. Large stretches of non-coding flanking sequences play a variety of regulatory roles that involve interactions with other molecules such as transcription factors. Most of the typical animal or plant genome contains vast stretches of non-coding DNA that can play a variety of useful roles, can serve as a genomic scrapyard, and also includes metaphorical selfish DNA elements, such as repetitive sequences that can expand, diversify, and move around the genome by a variety of mechanisms. Refer to Chapter 16. Other problems with the coding metaphor. The coding metaphor carries conceptual baggage that should be recognized as such and excluded from the updated evolutionary synthesis, UES. A code implies a code designer as well as a code breaker, and neither of these human mathematical or computer-assisted attributes apply to natural selection. There was no intelligent design involved in protein coding sequences in particular, nor in genomic evolution in general. This should in no way diminish the profound historical importance and brilliance of the creative conceptual leaps by Francis Crick and the pioneers who documented the detailed molecular genetics of the genetic code and protein synthesis. The quest to crack the code led to the current understanding that there is a many-to-one correspondence between the sequences of 61 of the 64 possible codons, DNA, RNA triplets, in protein coding sequences, and the 20 possible amino acids at each position in the sequence of a protein chain. Fortunately for the history of science and for our present-day understanding of biology, these pioneers made their discoveries largely because they were searching for a code. The metaphor was quite useful in that context. But it is time to move on to 21st century descriptions that represent a better approximation of the truth.